Good afternoon, everyone. My presentation of day two won't be quite as whiz-bang as Daniel's and Amber's, but we'll see how we go. Now, the speakers from day two, feel free to jump in. Now, I basically took copious notes yesterday and decided this morning that I can't present everything because it was a bit too much. So I've decided to take stuff out, but then I decided, well, not to do that because I might miss something. So what I've done is highlight stuff in red and green and other pretty colours to hopefully draw attention to it. So the day two report, really the focus being on innovation, big data, various OMICS, methods, computers, and in big red letters, collaboration. Don said this morning, and I agree, the answer's actually here in the room, whether it's for biomarkers, diagnostics, and of course candidate pathways for treatments. It's all here. You know, the knowledge is here. It's how we capture that. So collaborate, please. But before I do that, some of you are aware, there's a little bit of self-advertising. Under the Medical Research Futures Fund, there is a call to look at health economics in relation to ME/CFS in Australia. The ANU wants to put a proposal in, as I know other universities do, and we wish to establish an advisory committee. If you're keen, please see me, or if you want to contact me later, just go to the ANU website, put my name into staff. There's no other Lidburys actually in Canberra, let alone the ANU, and you'll find me quite easily. Send me an email. And the next thing I need to say, the advisory committee can be clinicians, of course, researchers, patients. I mean, people particularly like Emerge, representative advocacy groups. And just to remind too, as one of the two editors with Professor Paul Fisher, we have a special edition of Diagnostics coming out devoted to this conference. And we've also accepted papers from others who want to contribute by just an unsolicited paper submission to this journal, and we've had a couple accepted already. But this is going to be a bumper volume, I hope, and I said to Paul last night at dinner, like planting a flag, particularly for the biomedical thinking around ME/CFS and progress from this point onwards. So I can't force you, but particularly to the speakers, I'd love to hear from you, as would Paul, of course. We're just waiting like a wicketkeeper in cricket to catch those wonderful nuggets of wisdom, so please get involved with that. If you have time, as I said, by early May would be good. You know, we'll see how we go with that. But if you want to know more, if you just go to your search engine, just put in Diagnostics Journal, special issues, you can see more information about what's involved and how to submit your paper, which Paul and I will then assess. And basically, for those academics who you need to publish or perish, unless it's really awful, I mean, it's nudge, nudge, wink, wink, there's a pretty good chance it's going to get in, all right? So, hey, you know, what can I say? Sharpen those pencils. Okay, so session one yesterday, we were very privileged, of course. Thanks, Ron, for travelling as far as you have. Now, I just, as I said, I had to decide what to leave in and take out. It was interesting to hear Ron's comment, which reflected what Mark said this morning about GET, graded exercise therapy, is, I think the word was barbaric. We heard malpractice, we've heard some fairly strong language around that. The other thing that really resonated, because it agrees with what I've said to colleagues, is when you give funding, we do it, we fund the people who know what they're doing. There's always a risk through national research bodies. They say, great, we need to solve this, let's put out a general call, and all these people come in and take all the money, and often the people who have been in the field for a long time who really know it miss out because we don't have gilded CVs and amazing track records necessarily. So yeah, fantastic. We need that. So people to trust us and say, here's some money, get on with it. That'd be lovely. Scientists want to do science, essentially. Now, for the speakers, I might have already said this, just jump in if I get something wrong. DNA viruses have been looked at, a range of DNA viruses, nothing detected in Ron and colleagues' system. No new pathogens necessarily. We've heard about the risk of post-viral syndromes and other types of infections and pathogens. What I just want to flag here, you can just read the stuff in black, just read at your leisure. I'll make these notes available, but it's the red stuff that caught my eye, the multiplex cytokines, T-cell expansion, but of course, and this has come up with my work that I presented 2 days ago, is, yeah, we get these nice patterns, fantastic, but when you add more samples to the study, you no longer find the effect. So, and this is an issue that's been discussed a lot in Australia about we're doing at a discovery level, so at the biomedical discovery fundamental science level, some really excellent work that gets into good journals, but we need to scale up. We need many, many, many more research participants, and we need to do more work at actually validating our discoveries. and bringing together, again, all this knowledge in the room into something usable in the clinic, and which we heard about before, where actually I see myself as a translational scientist. All right. So just moving on, and I apologise to the speakers for just skipping over your notes. We had also in session one, Dr. Zhao, talking about a severe ME/CFS cohort, and I had to highlight this name here, Dr. Neil McGregor, of course, director of our nascent and emerging ME/CFS Discovery Research Network. And we're keen to collaborate, as Neil's already doing with Stanford and other colleagues around Australia and overseas. This was interesting, Dr. Zhao, if you're still here. There's been a lot discussed in Australia about Lyme, and Bruce commented on it this morning. I don't want to wade into this because people either love you or throw bricks at you, but it has been an issue about whether this is the cause, and it was interesting to see from that talk that no difference was found between ME and controls. This sort of thing rings my bell as a lapsed immunologist. The cytokine panels do show changes. 21 out of 61 changed significantly in the metabolite, 63 out of 592. For example, lysine. We heard a bit more about lysine with Neil McGregor's talk. So we got these interesting things going on. There are changes. So if you look at healthy, you know, again, what is healthy, but let's just put that to one side. A healthy research participant versus someone who fulfills whichever criteria and diagnosed as ME/CFS, there are differences. There's something going on at the metabolic and the immune level. I didn't quite catch the name of that gene, but anyway, we can fix that up later. The reason I'm rushing too, and I had this issue the other day, I was the last speaker before the wine tour, and today I'm the last speaker before lunch. So how to become really unpopular at a conference is cut into lunch or wine. Now, it was a great honour last night to hand out awards, well, to my friend and colleague Paul Fisher, but also to Dr. Fair, who is an electrical engineer by trade, and I often say that biologists particularly need to think more like engineers, and that is, rather than spinning around in circles, actually solve problems. So let's actually move it like it's an engineering project. So it's great. Thank you for injecting that can-do problem-solving approach. I didn't know about the Northfield Hospital in Adelaide and the ME/CFS epidemic there. I had no idea about that. Most of us know about the 1955 royal free disease in London, but I had no idea that we had such a history in this country. But what really, again in red, rang my bell, because other people from Ron Davis's network and elsewhere commented on this IDO2. What is that? And there's 5 common missense mutations, and there was some focus in this talk about the missense mutations being quite common. And this kynurenine pathway, did I pronounce that correctly? Okay. And the implications of also these pathways, which of course link immune system, gut function and brain, which of course are all implicated in ME/CFS. This bistability input versus output model, I didn't quite follow that, but please talk to Robert later if you want to know more. But what I did pick up too was, of course, the serotonin pathways. We talk about brain fog and various other, cognitive functions being impaired. So something very interesting there about the precursors, the phenylalanine, the tyrosines that Chris talked about as well, and dopamines and serotonins through these various sort of indole acetic acids and 5-HT hydroxytryptophan, sorry. Now this one, yeah, this is a dead set ripper, as we say here. A mean of 1.8 damaging IDO2 mutations in this IDO trap. Love to hear more about that, so I hope you submit a paper, Robert, because this is something that I think yesterday, or in day 2, the presentations from several of our overseas visitors really seemed to be a common theme about this IDO gene. And the kynurenine actual pathway, which was found to be decreased in ME/CFS. This seems quite compelling. Now on to our friend, who is from Melbourne and has moved elsewhere for the moment, Chris Armstrong. Thanks for your presentation, mate. The B-cell studies with the UCL team that was reported from day 1. Again, in red, increase in CD38. Now, for those uninitiated, I left immunology when we're up around CD 100. Now, there's about, I think we're up to about CD 250. They used to just call them things like TNF receptor 1 and 2, and I could handle that, but now it's look, I don't know what it does, but it's going up on B cells and increased in ME/CFS. And again, please talk to the experts. We've got plenty of them here. Very interested too, because I have some involvement here with the project at La Trobe with Paul, Sarah, Daniel and colleagues, and the greater utilisation of glucose in the media. There's something metabolically very different, and again talk to Paul, Daniel and Sarah about the cells that are extracted and grown in culture ex vivo from people with ME/CFS. They exhibit different things, and it is reflected, as we saw in Paul's talk, award-winning talk I must add, that it has a lot to do with the mitochondrial function, okay, and various complexes not operating as well as they should. Now, I just touched on it too with the whole thing about serotonin and the 5-hydroxytryptophans. We've got this thing going on with phenylalanines, which you must take in through your diet. Like you've probably all heard of PKU, you know, it's something you take in. And the production of dopamine. So again, looking at a neurological issue. So that was another study that Chris is involved in the metabolomics realm. Now, Ruud Remakers is a colleague from Radboud in Nijmegen in the Netherlands, and he got, probably because he finished early, but also he mentioned CBT, and there was a lot of discussion after Ruud's presentation. Are you here? No. Ruud actually did some work here with the Animal Health Laboratory in Geelong. He might be visiting them today, so it was just perfect that we invited him to Geelong so he could come here and also visit friends and colleagues. So the whole thing, which was, I mean, it's not something that's nice to talk about, but there was a massive outbreak in the southern Netherlands of Q fever, and a Q fever fatigue syndrome emanated in, I think he said 1 to 5% of patients who were diagnosed with Q fever, caused by Coxiella burnetii, I think named after Macfarlane Burnet, a great immunologist from Australia. Is that right, Paul? That's good. Yeah, Paul's Professor of Microbiology at La Trobe, so I always defer to him for these tricky questions. But also it was quite, as someone who spent some time actually at Nijmegen, I mean, let's face it, I know the man and was aware of the work over there, and part of the imprimatur of this meeting was to actually extend our invitations and form collaborations nationally, but also internationally, and I have connections over there. An expert centre in chronic fatigue and QFS has been going for 30 years, 40,000 cases, my goodness. Love to get hold of that data set. But the focus was on QFS, and it seemed from my reading that it was it's a fatigue syndrome, but it's a little bit different, or quite a bit different, in fact, to what we see. No persistent infection detected, and there was, of course, the mention of CBT, which did have some impact for the people soon after diagnosis, but then they relapsed. It was good that there was some mention of Professor Marmion, who's no longer with us, and his cytokine studies way back when, and the whole link with QFS and the Australian contribution to that. Again, as a lapsed immunologist, the immune responses, the pattern differences were quite compelling. You could see them just looking at many, many cytokines and other immune markers at once. You can see just through heat maps different patterns and how they shift. So there are differences at that level. There is an immune response, but it's different to what we see with chronic fatigue syndrome, ME. So it's interesting too, as a former virologist, that C. burnetii does replicate inside of monocytes, and I assume macrophages. And of course, I've highlighted here the link with mitochondrial function because of the downregulated genes. The mtRNR1 and 2 genes were found to be downregulated in their QFS sample. So very interesting too that there's mitochondrial proteins somehow affected in this case of fatigue syndrome post Coxiella burnetii or Q fever. Another interesting thing, the differences between the chronic fatigue and the Q fever fatigue was, we've talked a lot about the possibility of autoimmunity, and part of the evidence being the ratio, or 3 to 1 or 4 to 1 females to 1 male, but they were finding with manifestation of symptoms for Q fever fatigue syndrome of 50-50 male and female. So I think we could almost safely say, I mean, of course more validation is needed, that they're different diseases. Maybe that's helpful. Now back to Neil McGregor, who I mentioned before, who's got access to data from elsewhere, and I'm just checking the watch. Is it roughly 14 minutes to 1? Heidi? Oh, she's gone. She trusts me so much, she's not keeping time on me. That's great. So Neil is the most devoted biochemist. The sort of things he talks about takes me back to the late 1980s when I studied biochemistry and actually enjoyed it, unlike 98% of students who study biochem. So I have fond memories, but even so, I find it difficult. Pyruvate, lactate, fumarate, I mean, oh my goodness, my head's spinning. So there's lots of really good stuff going on. And of course, Chris, Paul Gooley, you haven't had much of a mention, Paul. Paul Gooley, stand up, round of applause. Hey. So the work done at Bio21 with our colleagues there involving Neil, Paul and Chris and Henry Butt and others, fantastic. And of course, you know, they've been working in this field for quite some time, again with Don Lewis at CFS Discovery. So, but Neil reported on, and again, someone interested in routine pathology data was very interested to hear about getting 777 cases, or it's actually more cases to start with, but cleaning the data and having that many cases to work with on hyperglycemia and actually stratifying the responses based on the GTT, depending on whether you had a flat response or you had different sort of kinetics in the insulin and glucose responses. And then looking at those relationships with anti-nuclear antibodies, serum creatinine, which again rang my interest because of what I reported the other day about the creatinine excretion rate over 24 hours for the patients in our study, and the difference, we could actually separate them based on how much creatinine, and in fact the creatinine, to answer the question, was actually lower, excuse me, significantly lower in the urine in our study. So there's something going on with the nitrogenous waste pathways, and there's some implications for urea as well. Neil talked quite a bit about what you see in hyperexercise or athletes, and the extension, I assume, Neil, being that, you know, you've got people with ME/CFS who are in an exhausted state, like maybe like what you might find after intense exercise, and what parallels are there. We do see electrolyte loss in our studies as well, but it's very labile result in terms of significance. The creatinine clearance through the urine is quite consistent, but the potassium and sodium rates of excretion in our data sets vary, sometimes significant, sometimes not. And as a lapsed molecular biologist, learning about the eukaryotic initiation factors was a very fascinating observation as well in terms of protein translation and maybe the disruption to that. That's normally impacted by viruses, which is another story. Yeah, I'll just pass that. The SMADs are interesting because I do some work on the activin proteins, and SMAD, the SMAD signalling actually is differential depending on which activin's involved, but I'll skip over that. Now, Dr. Moreau, thank you very much. The big thing here was the focus on post-exertional malaise and the microRNAs that were differentially expressed in these studies. And there's the list. So there's a range of microRNAs that have been found, and of course once you find these markers, whether it's a microRNA or it's a protein or it's a gene, you can then use the literature to actually understand what other scientists have found about their function and what they're involved with. So it's a good way to go because you can learn a lot from these experiments just by simply consulting the literature. So there's a few there. Would Dr. Moreau like to nominate which one is the golden key? He might not be here. Right there, all right. Which one?
I think it would be a panel of them.
It would be a panel. Good answer, actually. Yes. Yeah, that's right. There is no magic bullet. I mean, when I was learning science in the 80s and 90s, we were very obsessed, like antibiotics, with magic bullets. This will do it. This, like tumour necrosis factor was an area I worked in. This will kill the viral, you know, we'll make a magic bullet. No, it's about many things. So congratulations on that work. There's a number of candidates there that when you look at the various functions associated with these microRNAs, they do resonate in the context of CFS. Now, Dr. Bergqvist, again, welcome, all the way from Sweden. I was going to say, you put up ABBA, you said, what do you know about Sweden? As ever said ABBA, I was going to say IKEA. Anyway, so neuroimmunology, again, we're seeing regular reports about low-grade inflammation, again, flu-like symptoms, but again, just wanting to highlight, trying to pick up common themes was highlighted. And again, if I've got this wrong, please say, the mitochondrial involvement, and looking also at brain function. So I've just put little arrows here pointing up or down, just indicating, you know, whether, say, synaptic plasticity was increasing or axon repair decreasing. And the pregnenolone story, quite fascinating. That drew some attention and some comments from, I think, Bruce down the back there somewhere. Actually, I might go back. So the pregnenolone study, I mean, I learned a lot about that. I just thought it was a hormone and didn't think much more about it, particularly in terms of mitochondrial function and production. So amazing insights there. We had Suzanne McDonald speak about N=1 research, and I'm rattling through. Means misleading, and I just put a little note here that we do actually have a project running with my colleague Alice Richardson with Don Lewis from CFS Discovery, and one of the things we're finding, of course, is when looking at people with these differential pulse pressures, that you've got such variation that if you just do your standard means and standard deviation, the variation or the critical functions are being missed just by being lapped up by the variation and by finding a central tendency. So one patient statistically aggregated, and there can be interventions involved in these types of research strategies. And there was a paper published in, we discussed it yesterday, 2011 or 12, of what's called personal omics, where you just take lots and lots of data off the one individual over time. The question Paul Fisher had was, how is that different from longitudinal studies, or is it the same thing? So I'll let you two work it out over lunch. We're very lucky to have some computer scientists here talking about human-computer interfaces, and the way I'd link all of this together is to say that there's been a lot of talk just in the last couple of days here about measuring people when they have good days and bad days. And wouldn't it be great if we had some sort of app or some sort of computer interface where we could, just a bit like if you've ever had those 24-hour monitors put on you, some sort of monitor which we can actually, for those people who consent to that sort of research, we could actually track and actually see the variation in fatigue levels or some other measure. So the computer scientists are looking at this, and as they admitted, there's been very little done in this space in their discipline of computer science. But of course, with the technology nowadays, people carrying Fitbits and all sorts of technology on their wrist, wouldn't it be great if we could maybe combine that with Suzanne's N=1 research and collect lots and lots of data on individuals, and then come to the markers that we've been talking about and look at them as well, which might be a bit more challenging given that it's harder to collect blood away from the clinical environment. And just finally, the Euromean endeavour, which is part of a cost initiative through the EU. And Eliana spoke deliberately not about the science but about just putting together a network, which we're very keen to do, of course, in this part of Australia through what we call the MDRN. And we'd love to hear from people who are interested, particularly people who can help with recruitment of research volunteers and participants. What was interesting was how they've divided themselves into working groups, so they have this wide coverage. We tend in science to think, well, I'm a molecular biologist, I look here, I'm a neuroscientist, I look over here. But bringing those working groups together, I assume Eliana is about learning each other's languages and actually working together towards a common goal, which isn't easy. It was also quite interesting and alarming to hear that in some parts of Europe, MECFS just isn't considered, or you might have one practitioner in, was it Serbia or Croatia? Yeah, in those countries. Now that's, now what I've done as a final slide is, say collaborate. It's all here. We now need to put our arms out, grab it all and bring it into something meaningful that we can translate. Sooner the better. Okay. I'm a fundamental scientist. It used to be you just discover things and you publish them. I've moved out of that. It's now about discovering stuff and using them. You know, it's already lucky I have a career where I could do that for about 20 years, but you know, we need to actually think about the next steps. Discovery's great, but it's now talking to other people and moving it towards something. So the themes for me were the IDO, again, seemed quite fascinating in terms of it's consistent, as we heard from Robert and from Ron. And Xiao as well. The glucose tolerance, I found that a fascinating study, Neil, absolutely. And of course, how this fits in with amino acids, Krebs cycle, and all the things we tried to avoid as students at university because it was so hard to learn. But it's important. It's how we produce energy. The B cells, they keep coming around. We've had quite a B cell focus, both through the Latrobe work, but also from what we heard from UCL and colleagues there. And collecting the data through the human-computer interface, etc. data plus more data and good statisticians. That's something that we really need. We need to handle our data better and analyse it more deeply and more efficiently. And with Euromean, look, yeah, we'd like to be an associate. Our MDRN here in Victoria and the ACT and elsewhere, we'd like to be an associate one day. And just as my last slide, I'm just going to put up this out of a paper which you won't be able to read, but I just have to highlight some work that we did in at the ANU looking at using factor analysis to actually look at the different diagnostic criteria under the Canadian consensus criteria and what's most important. So if you're interested in that at a more deeper level, let me know, because I think, again, there's so much going on, it'd be really good to pull it back into something usable. So anyway, thanks for your attention. I finished early, but I'd like to, any discussions or questions? Happy. Thank you. No, okay. No questions.
Schedule's too tight, sorry. But in lunchtime, feel welcome to ask questions of the researchers and of Brett. So I just have a couple of things to say. The first one is that for everyone that would like to join a round table, so if you are not a keynote speaker and you would like to join a round table, it is essential that you give your name to Georgia on the registration desk over lunchtime. So if you came in and you registered and you saw Georgia write your name down in her spreadsheet, then you're going to be assigned. But if you can just check that your name's on the list, because some people were already registered and you've not been picked up. So if you could make sure that happens. Lunch is going to be from 1 p.m. until 1:40, what time is it now? 1 minute to 1. Okay, we're keeping to time. This is good. So the other thing that I want to do in the 1 minute that I have left is, as the people who were here yesterday saw that we did a Best Presentation Award for Day 1 and a Best Presentation Award for Day 2, which went to Professor Paul Fisher and to Dr. Robert Fair. We also had, so we've been, this conference has been partly sponsored, or that's not the right word, is it? Diagnostics Journal are doing a special MECFS journal, which we're encouraging all of our speakers to submit papers to. And as part of their support for the conference, the Diagnostics Journal provided a prize for the 2 best presentations. Now, there was also a prize for the best presentation on day 3, but because of the format of the programme, it didn't really work. So what Diagnostics would like to do and have asked us to do is we want to partially recognise the up-and-coming emerging researchers in this field who've been in attendance here and have been helping us out immensely, which is Amber J. Quay, Jacqui every time, and Daniel Missalidis. So it's a thank you, and it's a recognition of their work emerging into this field. And if you guys can come up to the front, you're going to hold the certificate and get your photo taken. And it comes with a $50 gift voucher each from the Diagnostics Journal. So I sort of hand you this. You have to hold it here, and we get the photo taken. We need to see the diagnostics. Where are we? And now it's lunchtime, back at 1:40. Thank you.